Hello there, class. So good day to all of you. So for this pre-recorded lecture video, we will be talking about the nitrogen metabolism. So in the past few modules that we have, we already discussed the metabolism of your carbohydrates and your lipids. So the other element that is crucial in the biomolecules natin is your nitrogen. So in this lecture video, pag-usapan natin yung mga nitrogen-containing compounds natin and paano ba siya nagagamit sa ating katawan. Okay? So the first question that we have here is, where do we get that nitrogen? So that nitrogen comes from the atmosphere. So the atmosphere contains at around 78% of nitrogen gas, and that occurs as an N2. It's a diatomic molecule. So that nitrogen gas is not readily used up by our organisms. So we have here a link between this unutilized nitrogen gas, and it becomes available for our organisms. So the first thing is we use the process what we call your nitrogen fixation. Okay, so nitrogen fixation, and there are two ways in which the nitrogen fixation occurs. And why the way? What is this nitrogen fixation? So nitrogen fixation is where is the process wherein you convert your nitrogen, your N2, into NH3 or NH4 plus. Okay, so we have two ways on how to produce this ammonium or ammonia. The first one is via nitrogen fixation in the root nodules of legumes or yung mga leguminous plants natin. Meron doon sa nodule yung mga parang inflamed portion ng mga roots, meron doong nitrogen fixing bacteria. Okay? The other one naman is if you have this bacteria that contains a nitrogenase enzyme. So dito sa part na to. Okay? Okay, so assuming that we have now your ammonium, this ammonium will be further magkakaroon ng mga reactions pa. So we call that your nitrification, wherein you convert your ammonium, your NH4 plus, into nitrates. And then there's another reaction wherein the nitrifying bacteria will convert these nitrites into nitrates. And we all know that these nitrates are the ones that are readily used up in plants, diba? So ito yung nagserve as fertilizers natin. Okay? So ito yung nagserve as fertilizers natin. And the plants do assimilate this. When say assimilate, ginagamit ito ng mga plants to produce your amino acids and of course your proteins. So these plants are eaten by your animals, diba? We all know that in the ecological perspective that plants are the primary producers natin, and we are the consumers of these plants. Now, when we die, so when organisms die, so namamatay sila, these plants and animals are acted upon by your decomposer. So we have decomposition activity. So with the help of aerobic and anaerobic bacteria and fungi. So na yung mga nitrogen sa katawan ng plants and animals natin will be magproproduce siya ng ammonium. Yon. So also, aside from that decomposition activity, yung ating mga animals din ay nag-release din sila ng waste products. So we humans, we are mammals, we excrete urea. So urea is another nitrogen containing compound that is released kapag tayo umiihi. So may iba naman na like fishes, nag-release naman sila ng diretsyo ng ammonia. Okay? So ammonia yung nire-release nila. And some birds ay nag-release naman sila nung tinatawag natin na mga guano. Okay? So now, where's the continuation of that story for the nitrogen utilization? So, assuming that we have now the decomposition, then we have the formation of your ammonium. So, again, this ammonium will be converted into nitrites, by a certain bacteria. So we have this the nitrifying bacteria, and then another set of nitrifying bacteria will convert the nitrites to nitrates, and these nitrates can be acted upon by this the nitrifying bacteria to produce your N2 gas. So binabalik natin yung naibabalik yung nitrogen in the form of nitrogen gas. Okay? So that is the whole idea of the nitrogen cycle. Okay. So the nitrogen metabolism in animals. So nitrogen enters the body as amino acids contained in the dietary protein. So we do consume food, and yung food natin ay may kasama yung mga amino acids or proteins. And this nitrogen leaves the body as urea. It can also be in the form of ammonia and some other products. Okay? So urea is our main excretion product in na nitrogen containing. Okay? And ang ating mga amino acids and proteins, this is the third in rank for the energy source. So we do all know that carbohydrate is the first in the rank, and then after that is your lipids. So if your carbohydrates and your lipids are all used up, then we use your amino acids or proteins for source of energy. So here in this slide, we have here the overview of the protein metabolism. So your dietary proteins will be degraded into the component amino acids. So you all know that we have your mga peptidases, okay? So mga proteases natin. Also, some intracellular proteins, yung mga proteins na sa ating katawan mismo na nagagawa natin, that can also be degraded, and some amino acids can be used from yung dietary proteins natin na na-degrade to amino acids can be used to build up the proteins that we need. So, di ba, we need a constant supply of enzymes, di ba? So, doon napupunta yung mga amino acids natin. Now, if you recall the structure of your amino acids, we have this what you call your alpha amino group that is bonded to the central carbon, which you call your alpha carbon, which is in turn bonded to an H or hydrogen, and then your alpha carbon is bonded to this alpha carboxyl group, and we have this variable group which is called your side chain. So yung ating amino acids na yan, we can split that into two. So the first one is what we call your amino group. This amino group, the NH3 plus, the NH3 plus here will be excreted. So yan, pwede siyang ini-excrete, or the NH4 here can be used for the biosynthesis of amino acids, some nucleotides, and biological amines. So ano yung tawag dun sa natirang portion ng ating amino acid? So this part is what we call your carbon skeleton. Okay? So this carbon skeleton can be used up for itong part na to, pwede siyang gamitin dun sa citric acid cycle. So pwede siyang pumasok dyan. And some intermediates that stem or nanggagaling dito sa ating carbon skeleton can be used up for the process of gluconeogenesis. So gluconeogenesis is the synthesis of glucose from non-carbohydrate intermediates or precursors natin. And also, as part of the citric acid cycle, we remove your carbons in the form of carbon dioxide. We also produce water, and if the cycle goes on from Krebs cycle, that will enter the electron transport chain to produce your ATP. So the ATP here is the source of your energy. Okay. So now, imagine that all your proteins are degraded, your dietary proteins are also degraded. We have this what we call your amino acid pool. Okay. So the amino acid pool contains all the amino acids that are distributed all throughout the body. So naiipon, if you recall our discussion on the cholesterol, yung, di ba, ganun din siya, naiipon yung certain product na yon sa isang organ. Okay? So, naiipon din. So, itong mga amino acids natin ay naiipon din siya. Okay? So, ano ba ang maaaring paggamitan nitong mga amino acids natin? So, this will be the fate or kung saan mapupunta yung mga amino acids natin. So, we already mentioned that your body proteins are degraded and that will go to the amino acid pool. And same goes with your dietary proteins. Yung mga kinakain natin ng mga pagkain, may proteins yan. So, i-chop sila, tapos madadagdag sila sa amino acid pool. Okay. So, meron din tayo ng mga non-essential amino acids. So, kung natatandaan nyo pa, yung discussion natin ng non-essential amino acids, these are readily synthesized by our body. But we do have your essential amino acids which should be supplied in the diet. So, yung natatandaan pa ba yung mnemonics natin for essential amino acids? So, we have there the private math hill. Okay. So, ngayon, meron ka na ng amino acid pool. So, saan ngayon mapupunta itong mga amino acids na to? So, we have now your synthesis of your proteins. So, may mga proteins kasi na after some time na magamit ay dinidegrade sila. So, pag kailangan mo na ulit siya, sinisynthesize mo naman siya. Okay? The next portion is you synthesize your other nitrogen-containing compounds like porphyrins, purines, and pyrimidines. So, if you're familiar with purines and pyrimidines, these are compounds are associated with your nucleic acids. Okay? So, yan ay ginagamit for the biosynthesis ng ating nucleic acids. So, you know, nucleic acids, these are your DNA and your RNA. Then, as I mentioned earlier, in the previous slide, your amino acid, the carbon skeleton, can be used up in other pathways or it can be burned as fuel. So, I mentioned that it can be, pwede siyang papuntahin sa gluconeogenesis so that you can create your glucose. O kaya naman is you burn that as a fuel. So that when you say burn it as a fuel, then you allow it to enter the Krebs cycle and then the electron transport chain. Okay? So again, what are the sources of amino acids natin? So you have your amino acid biosynthesis. We distinguish yung amino acids natin as essential versus non-essential. So essential, you need to supply this in your diet. Yan, sinusupply natin yan sa diet. While your non-essential, we can synthesize this inside our body. So yung isang characteristic ng essential amino acids ay yung mga aromatic amino acids natin. So medyo mahirap kasi i-synthesize yung ring ng mga amino acids na aromatic like your tyrosine, your tryptophan, and your phenylalanine. So, kaya kailangan silang i-synthesize. Kailangan silang i-source out sa diet natin. So, the other source of amino acid will be the protein degradation in the body. So, this is part of the protein turnover and also the intake of your dietary proteins. So, paano ba yung paano yung ways kung saan natin pwedeng magamit yung mga amino acid natin? So, yung mga amino acid natin kasi, when say biosynthesis, we create your amino acids. So, yung carbon skeleton, nanggagaling siya dun sa mga metabolic intermediates natin. So, and, like for example, ito, yung ating pyruvate, yung pyruvate natin, if we do some little reaction on that, we can produce your alanine, your valine, your leucine, and isoleucine. Now, kung 'di ba ina-assemble natin yung structure ng amino acid mo, that you have your amino group, the alpha amino group, and the carbon skeleton. So, yung carbon skeleton manggagaling dito sa mga to. Okay? Tapos, yung amino group, the NH3 group, yan ay pwedeng manggaling sa ibang amino acid. So, minsan, ibinibigay o pinapasa yung yung alpha amino group from one amino acid to another. Okay? Also, we have your protein turnover. So most proteins in the body are constantly being synthesized and then degraded. So when the need arise, kailangan siyang i-synthesize. So pag hindi na kailangan, saka siya dinide-degrade. So the degradation of these proteins will permit the removal of abnormal or unneeded proteins. Kasi minsan nagiging problem yan with metabolism that some proteins do become non-functional o nagiging misfolded siya. So, as I've mentioned earlier, lagi ko naman na-mention na kapag yung mga proteins natin ay nagiging non-functional, may profound effect siya sa body. Okay? So, kailangan nating maiwasan yung mga abnormal or unneeded proteins na yan kasi baka magkawan siya ng medical na sakit. Okay? So, paano natin maiiwasan o paano natin i-degrade yung mga proteins na hindi na natin kailangan? So, we have two mechanisms. So, the first one is we degrade your proteins, the unfolded, non-functional, wala nang silbing mga proteins, by enzymes in the lysosome. So, tanda pa ba ang ating cell structure? So, yung lysosomes natin, they contain hydrolytic enzymes. So, hydrolytic. Ang ginagawa nila, kinakat nila o china-chop-chop nila yung mga biomolecules doon sa lysosomes. And the other one is the most famous is what we call your ubiquitin proteasome mechanism. So, paano ba yung ubiquitin proteasome mechanism na yan? So, yung ubiquitin, it serves as a tag. So, di ba? Ayan, sabi dito, ubiquitin tags proteins for destruction. So, pag parang it's an indication that if the protein is being tagged for destruction, so yung ginagamit doon is the ubiquitin. Okay? So paano ba nangyayari yung tagging ng inyong proteins? So your proteins do contain lysine. So may mga for sure naman na present naman ng lysine sa inyong protein, yung protein na gusto mong i-degrade. So, yung lysine, doon ikakabit si ubiquitin. So, the more lysine your protein contains, the more lysine dun sa yung protein, so mas madaming ubiquitin yung mas mailalagay. So, pag indication na pag madaming ubiquitin o may ubiquitin na nakalagay doon sa protein mo, then it is destined na ma-degrade siya. Okay? So, sino yung magde-degrade sa kanya? So, yung tinatawag natin na proteasome. So, the proteasome digests the ubiquitin tag proteins. So, yung itsura niya, mukha siyang ganito. Meron siya nung cap, meron siya nung catalytic subunit, tapos may another cap. So, yung structure kasi nito, meron siyang parang hollow portion. So, yung may butas yan sa gitna, na dyan, pumapasok yung mga proteins na natag with ubiquitin. Okay? So, to illustrate, this is yung itsura niya. So, yung proteins natin na destined to be degraded, yan ay nakabitan na ng ubiquitin. So, nakatag na siya. Then, this will proceed or this will go yung proteins natin dun sa inyong proteasome. So, yung mangyayari ay yung proteins ay parang siyang imagine nyo yung shredder, paper shredder. So, di ba pag sinabi natin, ito, hindi na to kailangan, so itatapon to. So, i-shred mo na yan. So, parang ganun yung mechanism niya. So, the proteasome here acts as a shredder. Okay? So, yung protein mo, pinifeed mo siya doon sa loob ng proteasome, and ito yung parang cross-section niya, or, yung cut section niya. So, the proteins are being pinifeed siya dun sa loob ng proteasome. Okay? So, yung mga ubiquitin na nakatag ay naaalis din siya. Naaalis din siya dun sa pagkakatag dun sa protein. So, anong ginagawa ng proteasome? So, yung proteasome, para siyang meron siyang parang ngipin sa loob. Yun yung parang simplest explanation na kinakat niya, parang minamunch niya, nginangata niya ng dahan-dahan yung protein mo. So, pagkatapos doon ay ito na. Kaya siya hollow yung structure ng inyong proteasome kasi kailangan makalabas yung chinop-chop mo na protein. So, dito siya lalabas sa other side or the other end of your proteasome. Okay? So, itong mga, ano pa yan, kasi hindi pa totally degraded. Hindi pa totally degraded itong mga proteins natin dito. So, meron pa itong mga series of other reaction na kung saan pwede mag-akka pa naman yung mga enzymes natin, yung mga carboxypeptidases natin, and mga enzymes for proteases in general. Okay? So, yan. So, pag yun ay na-cut pa siya ng further into smaller units sa amino acids na siya, then pwede na siyang mapunta dun sa tinatawag natin na amino acid pool. Okay. So that is one side of the story. Now, we do have here yung introduction ng proteins sa ating diet. So we have your dietary proteins. So let's discuss a little bit of the digestion and the absorption and transport. Okay. So allow me to share this picture with you. So of course, pag digestion, ang system na involved dito is your digestive system. So, in the mouth, there is no digestion of proteins. Bakit? Kasi dito pa lang nagkakaroon ng mechanical breakdown of food into smaller particles. So, minamunch natin, minangata natin na nagiging smaller particles yung food natin. Okay. So, it will dahan-dahan siyang dadaan sa esophagus, tapos ang susunod niya ma-encounter is your stomach. So, we all know that in stomach, we have here your hydrochloric acid. So, yung gastric juice natin. So, yan ay acid. So, the tendency is that hydrochloric acid will denature your proteins. Okay? So, yan. Na-denature yung proteins natin. And, when say denaturation of proteins, nawawala na or naaalis na yung kanyang 3D structure. Yung kanyang folding. Also, in the stomach, we have your pepsin. So, this one is an enzyme. Yan. So, this helps in hydrolyzing the peptide bonds to form large peptides. So, kinakat din yan ang pepsin. So, the next one is it will go to the small intestine wherein it will encounter your trypsin, your chymotrypsin, carboxypeptidase, aminopeptidase. So, your trypsin and chymotrypsin here are pancreatic proteases. So, itong trypsin and chymotrypsin, this comes from your pancreas. Okay? And, itong natira natin, yung carboxypeptidase and aminopeptidase, these are blood barrier enzymes. Okay? So, ang effect lang nila, tulong-tulong kasi yung mga yan sa pag-degrade ng inyong pag-degrade ng inyong proteins into smaller fragments, into amino acid. So, they hydrolyze the peptide bonds to form your amino acid. So, majority of the digestion and the majority of the digestion happens in your small intestine. Okay? So, kapag maliit na, naging amino acid na lang siya, these amino acids will be absorbed. So, i-absorb yan ng ating intestinal lining. So, kung natatandaan nyo, yung ating yung structure ng small intestine, meron tayo ng villi. So, yun yung parang mga yung cute na structure. Kaya ganun yung structure ng villi kasi it will increase the surface area. Kasi kung yung ating small intestine ay ano lang siya, tuloy-tuloy na smooth lang siya, maliit lang yung area. So, para mas madaling ma-absorb yung ating mga biomolecules. So, the idea here is, meron na tayo mga free amino acids, and these amino acids will be transported to the bloodstream. So, the intestinal mucosa will absorb the amino acid. So, pagkatapos nun ay itatransport na siya. Okay? So, ito lang yung parang bigger picture niya. So, the amino acids of the protein fragments, yan. So, dahan-dahan na siyang dahan-dahan na siyang ika-cut. So para maka-form ka na ng um, maka-form ka na ng amino acids mo. I stand corrected with sa sinabi ko kanina na itong carboxypeptidase and aminopeptidase, these are, kasi sa notes ko, BB nakalagay. So these are your brush border enzyme. So I stand corrected. So, yan. Ngayon, meron tayong absorption na mangyayari dito sa inyong epithelial cell. So, ang magpapapasok noon from the lumen of the intestine papasok dun sa ating epithelial cell is what we call your sodium-dependent transporter. Okay? So, papasok na siya dyan. Tapos, this epithelial cell, meron siyang adjacent na capillary. So, palalabasin mo siya, padadaanin mo lang siya. Yung amino acids mo, padadaanin mo lang doon sa epithelial cells mo para makapasok siya sa bloodstream. Okay? So, capillary lagi yung unang pinapasokan kasi we know that the capillary is the smallest blood vessel. Then, as yung distance ay lumalayo, di ba, lumalaki yung blood vessel mo, nagiging arteries. Yon. So, what is responsible for the transport ng inyong amino acid from the epithelial cell papunta sa bloodstream is what we call your amino acid carrier. So, this amino acid carrier is sodium-independent transporter. Kasi pag sinabi mong sodium-dependent transporter, para makapasok yung amino acid doon sa epithelial cell, kailangan mo ring magpapasok ng sodium ions. Pero pag dito na sa portion na ipapadaloy mo na yung amino acid mo sa bloodstream mo, hindi na kailangan ng sodium. Okay? So, we discuss now the protein and amino acid catabolism or degradation. So, ano ba yung tatlong situation kung saan nangyayari itong catabolism? So, number one is when the dietary protein is more than the proteins needed for the synthesis of body proteins. So, lagi lang tayong meron lang tayong cap. Meron tayong optimum level of proteins na kailangan sa body natin. So, anything in excess will be degraded. So, that's the idea of number one. Number two is when during extended period of starvation, when glycogen and lipid reserves are almost depleted. So, at the start of this lecture, I had mentioned that your carbohydrates will be the first in the priority for the source of energy, and then second natin doon is yung mga lipids natin via the beta oxidation pathway. Pero kapag wala na talaga yung dalawang yon, ang susunod na natin gagamitin is your proteins and your amino acids. And the last one is what we call your protein turnover. So this happens with your continuous synthesis and degradation of your body protein. So kapag kailangan ng proteins, you synthesize it. Kapag hindi na kailangan, saka mo siya i-degrade. Okay? So that's the idea of protein turnover. So, yun nga. So pwede mong gamitin yung amino acids mo for the synthesis of your body proteins. Pwede mo siyang gamitin for the synthesis of your precursors or starting materials for your nucleic acids, and the carbon skeleton can be used for other pathways or pwede siyang gamitin na a source of energy. So we burn it as a fuel. So again, this is just an overview. Na-discuss ko na to kanina. So yung gusto nating puntahan is when you have your amino acid, pwede natin siyang i-excrete yung amino group in the form of urea, and yung carbon skeleton naman is pwede natin gamitin for the synthesis of glucose via the gluconeogenesis pathway, or we can use that, we allow it to enter the citric acid cycle or the Krebs cycle. So ito yung aerobic respiration. So mag-produce yan ng carbon dioxide and water. And yung nawala dito is the production of the ATP in the electron transport chain. So ito, this is yung sinasabi ko na kanina, yung ating structure ng amino acid. We dispose the nitrogen atom in the form of urea. And the remainder of the structure of your amino acid will be used for recycling. So, when you say recycling, ito nga ay pupunta sa energy o kaya naman ay pwede natin siyang gamitin to synthesize other organic compounds. Okay. So, nung simula ng semester, nasabi ko na sa inyo na napakaganda nung discussion ng biochemistry kasi meron tayong mga ways, meron tayong methods kung paano natin dispose yung mga unwanted materials or products or unwanted substances sa ating katawan. So, we have here the excretory forms of nitrogen. So, most aquatic vertebrates like fishes, nire-release yung nitrogen in the form of ammonia or pwede rin siya ng ammonium ion. Okay? So, in our case, tayo ay mga mammals, tayo ay kabilang sa terrestrial vertebrates, so we excrete your nitrogen in the form of this urea. Okay? And yung birds and reptiles naman ay ini-excrete nila o nire-release nila itong nitrogen in the form of uric acid. Okay? So, may complexity dun sa structure ng ating, sa chemical structure. Yan. Depende yan sa kung anong species yung pinag-uusapan natin. Okay? So, we first go to the disposal of your nitrogen. So, yung nitrogen nga natin ay kailangan natin siyang i-dispose kasi ang ang ammonia, kapag yan ay naiipon sa katawan, that is delikado din yan. Kasi di ba ang ammonia isang base yan. So, as a base, it will have an effect dun sa pH level ng ating blood. So, kapag may ammonia, so that's a base, then we have your alkalose. Makaka-contribute yan sa imbalance ng pH natin. So, we need to remove your nitrogen. So, we have 3 ways on how to remove your nitrogen in your amino acid. So, the first one is what we call your transamination reaction. And the second is oxidative deamination. So, deamination. So, meron na kayong idea dito na deamination, when a term starts with de, it means you remove. Okay? You remove. Pag transamination naman, lagi tayong nasasanay na yung trans ay, ang ibig sabihin nito ay tinatransfer natin siya. Okay? So, tinatransfer natin yung amino group. Yung isa naman ay directly, we remove the amino group. And the third one is the most relevant of all is what we call your urea cycle. Okay. So, let's discuss first your transamination reaction. So, in the transamination reaction, this occurs in the liver cytosol. So, bida na naman yung liver natin. So, lagi kasing, pinakamahalaga, the most important metabolic organ in our body is your liver. So, dapat kailangan mapangalagaan nyo yung liver nyo. Kasi majority of the metabolic pathways happen in the liver. So, what happens in a transamination reaction? There is a transfer of the amino group to an alpha-keto acid. And that is usually our alpha-ketoglutarate. So, yung alpha-ketoglutarate, ito ay na-encounter na natin sa Krebs cycle. Okay? So, sa Krebs cycle. So, siya yung tatanggap ng amino group. Okay? So, the name of the enzyme is usually called a transaminase or an aminotransferase. So, yung dalawang yan ay synonymous lang siya. So, dun sa reaction na yan, kapag nalipat yung amino group ng isang amino acid at tinanggap siya ng alpha-ketoglutarate, the alpha-ketoglutarate will result to the formation of your glutamate. So, di ba, ang glutamate natin is NH2, C, H, COOH, and then we have CH2, CH2, and then COOH. So, yung part na to, pag wala tong yung amino group na yan, this is what we call your alpha-ketoglutarate. So, pag nalagyan siya, nagiging glutamate na siya. Okay? So, let's give an example. Yan. So, ito lang yung parang pictorial version niya. The amino acid 1, this will be the source of your amino group. It will be donated to the alpha keto acid. Okay? So, that will be acted upon by your aminotransferase or the transaminase enzyme. By the way, this enzyme will require a pyridoxal phosphate which serves as a prosthetic group of your enzyme. And the pyridoxal phosphate comes from the vitamin B6. Okay? So, at the end of the reaction, magkakaroon kayo ng formation ng alpha keto acid. So, itong amino acid na yan, naalisan siya ng amino group. So, magiging C double bond O yung carbon dyan. And then, the amino group from the amino acid number 1 will be transferred dito sa alpha keto acid natin, which is usually your alpha keto glutarate. So, the alpha keto glutarate will become now your new amino acid. And this one is usually in the form of your glutamate. Okay? So, may relevance bakit glutamate yung ating bakit glutamate yung parang doon kasi sa glutamate doon mako-concentrate yung amino groups ng ibang mga amino acid. Okay? So, ito lang yun siya. So, ano yung mga pwedeng tumanggap ng inyong amino group from an amino acid? So, the first one is your pyruvate. So, pyruvate is the end product in your glycolysis. So, if you add here, an amino group, so magiging ano siya? Yung magiging structure niyan is magiging alanine. Okay? So, alanine natin is just CH3 yung side chain. Okay? Your oxaloacetate can also accept this NH3, the amino group. So, the oxaloacetate, pag nagkaroon tayo ng transamination reaction, it will become your aspartate. And then, the alpha ketoglutarate nga, so ito yung usual na ginagamit natin. So, if you add the amino group in this portion, this will become the structure now of your glutamate. Okay? So, ito lang yung sinasabi ko kanina. So, yung alanine, siya yung magdodonate. So, siya yung magdodonate nitong, siya yung source ng amino group, and this will be transferred to the alpha ketoglutarate. Okay? So, what happened? The amino group is transferred, naging glutamate siya. So, yung alanine natin ay naging pyruvate. Okay? So, maganda yung system na ganito kasi ang gusto kasi natin ay matanggal yung amino group kasi ngayon ay for waste production. So, waste product na siya. So, kapag nangyari itong transamination reaction, na-produce natin yung glutamate, yung pyruvate. So, your pyruvate can be used for source of intermediate. Siya yung pwedeng pumasok saan? Pwede siyang pumasok sa tricarboxylic acid cycle nyo or the Krebs cycle. Okay? The other one is in the reaction of your aspartate with your alpha ketoglutarate. So, si aspartate naman, siya yung source ng amino group natin. It will be transferred to alpha ketoglutarate to produce your glutamate. Okay? Ngayon, ano nangyari kay aspartate? Si aspartate, nung natanggalan siya ng amino group, it became oxaloacetate. Okay? So again, ano na naman itong relevance ng oxaloacetate? The oxaloacetate here is part of your Krebs cycle. Okay? So, sana na-realize nyo na ngayon na kapag kulang yung intermediate sa ating Krebs cycle, pwede natin gamitin yung mga amino acids natin for the formation of, for example, oxaloacetate. Diba? Remember, in the Krebs cycle reaction natin, the oxaloacetate will condense with your acetyl-CoA and that will produce your citrate. So yung acetyl-CoA na yan, hindi siya makaka-proceed with the Krebs cycle kapag kulang yung oxaloacetate. So, para magkaroon tayo ng oxaloacetate na supply sa Krebs cycle, kailangan mo nang ma-produce yung oxaloacetate. So, one way to produce that is via the transamination reaction of your aspartate. Okay? So, ano siya, parang dalawang, ano siya, yung nabanggit ko nga, dalawang benefit niya. Matatanggal natin yung amino group, may excrete natin yan. Tapos, pwede natin gamitin yung mga na-produce natin, yung mga products natin as intermediates or material sa ating Krebs cycle. So, the second one is through the oxidative deamination. So, this again occurs in the liver and in the kidney. So, ang idea dito ay nagtatanggal tayo ng amino group dun sa glutamate. Kaya gusto nating ma-produce yung glutamate dun sa transamination reaction kasi pag naipon yung glutamate, tatanggalin na natin yung amino group na yon, di ba? So, tapos, pag natanggal natin yung amino group, ay na-recycle natin yung alpha ketoglutarate. And we know that in the previous discussion, the previous slides natin, that alpha ketoglutarate will be the one to receive the amino group of all other amino acids. So, magagamit natin siya. Ma-recycle natin siya for another round of transamination. Okay? So, the site niya, yung actual site ng oxidative deamination is in the mitochondrial matrix. Okay? So, another relevance nga ng alpha-ketoglutarate for transamination o kaya naman kapag kulang na yung inyong Krebs cycle intermediate na alpha-ketoglutarate yan. So, pwede mo tanggalin yung amino group ng glutamate. Okay? Yan. So, here's an example of your reaction. So, the glutamate, so sa kanya naiipon lahat yung amino group na tinanggap niya from other amino acids. So, with the aid of water, nagkakaroon tayo ng hydrolysis. So, naaalis natin yung amino group na nakakabit dyan and we use here NAD+. Okay? So, aalisin natin yan. It will the glutamate will be converted back to alpha-ketoglutarate. So now, meron na tayo nung free na NH4 or the ammonium ion. So ngayon, ano na yung fate nitong NH4 na to? So this will now go to the third step. Okay? So yung ating tatlong steps kanina dito ay connected sila. Yan. Connected silang tatlo. So the last step is via the urea cycle. So, yung NH4, yan ay papasok na sa urea cycle. Okay? So, yung gamit natin na enzyme pala dyan is the glutamate dehydrogenase. So, kasi, pag meron tayong NAD+, laging dehydrogenation reaction yan. Okay? So, what is this urea cycle? So, urea cycle is the one responsible for the detoxification of your ammonia or the NH4 plus from your amino acid degradation. So in this process, we convert your NH4 plus, your ammonium ion, to urea in the liver. So meron tayong cellular localization nitong mga reactions. So yung first two steps will happen in the mitochondria. Kasi nga 'di ba nangyayari yung oxidative deamination inside the mitochondrial matrix. So, doon muna siya mangyayari. And then, the rest will happen in the cytosol. Okay? So, urea, ito yung structure niya. So, it is the major form of it's the major disposal form of amino group natin, especially humans. And this accounts for the 90% of the nitrogen-containing compounds in urine. So, may iba pang mga substances na e-excrete sa urine natin, but the 90% of nitrogen-containing compounds will be in the form of urea. Okay? Kaya minsan gagamit yung urea, yung mga ihim, if you know agriculture, Indian agriculture, so yung urea, yung ihim ng baka, minsan ginagamit siya for a source of a source of ginagawa siyang pandilig. So, essentially, para siyang fertilizer. But, minsan, namamatay din kasi yung halaman kapag sobra naman yung supply. So, dapat meron lang laging optimum level or optimum na isusupply. Okay? Sa inyong mga plants. So, here is the overview of the reaction. So, the first two steps will happen in the mitochondrial matrix and the rest will happen in the cytosol. So, itong box na to, parang ito yung, imagine this is the mitochondria. Yan. Tapos lalabas siya, cytosol. And at the end of the reaction, we have the formation of your urea. Okay? So, the first step is, yun nga, sa mitochondrial matrix. Ayan. And the rest will happen in the cytosol. So, mamaya ko na i-discuss itong relevance ng maliit aspartate shuttle. Medyo na iba yung sequence. So, some urea cycle intermediates natin. So, maririnig nyo ito mamaya. We have your arginine. That is one of your amino acid. The next one is ornithine and then citrulline. So, arginine is one of the standard amino acid natin. And the citrulline and ornithine, these are your uncommon amino acids natin. Okay? So, pasok pa rin sila dun sa criteria na amino acid. Pero hindi ito kasing mga to ay hindi ginagamit for the synthesis of your proteins. So, yun yung tatandaan nyo. Yung 20 standard amino acid versus the all other uncommon non-standard amino acid. Kasi 'di ba, ang amino acid natin, ang basic structure niya is you have the amino group that is bonded to the alpha carbon, and that alpha carbon meron siyang alpha carboxyl group, and then meron kang H, and then the rest is your side chain. So pasok pa rin sila doon sa criteria na yun. Pero yun nga, hindi ginagamit for the synthesis of your proteins. So, what happens in the reaction? Yung ating NH4 that comes from the glutamate, that will react with your carbon dioxide. So, your carbon dioxide here is another waste product. Okay? So, waste product natin siya, and with the help of water. So, you need here ATP. So, di ba parang yung mga waste products mo ay gagamitan mo pa siya ng energy. Okay? Para makapasok kasi para makapasok sa urea cycle, kailangan muna siyang ma-convert into this compound which we call your carbamoyl phosphate. Okay? So, yung carbamoyl phosphate na yan ay galing sa yung NH2 comes from your glutamate. This part comes from the carbon dioxide, and meron tayong nagamit na phosphate. So, yung dalawang ATP natin ay nagamit siya. So, di ba supposedly meron tayong pag nag-hydrolyze ka ng dalawang ATP into ADP, so dapat may isang ADP, isang phosphate tayo. Dahil ito dalawang ATP, nag-hydrolyze siya, naging 2 ADP, yung isang inorganic phosphate lang yung natira. Kasi yung isang inorganic phosphate ay inilagay dun sa structure. So, this is now your carbamoyl phosphate. Okay? So, kapag na-produce na yung carbamoyl phosphate na yan, saka na magko-commence yung ating urea cycle. So, cycle ito ha. So, katulad dun sa Krebs cycle, makikita nyo mamaya na meron tayong portion na ma-regenerate natin yung isa sa ating mga intermediate doon sa urea cycle. So, dahil na-produce na natin yung carbamoyl phosphate, that will condense with your ornithine. Okay? So, para siyang katulad nun sa Krebs cycle na ang intermediate na nandun laging available is the ornithine. Tapos, ang papasok lang is yung itong carbamoyl phosphate. So, yung ornithine dito, at the end of the reaction, ma-regenerate natin siya. So, what happens? Your ornithine will condense with your carbamoyl phosphate to form this citrulline. So, citrulline is, one of our uncommon amino acid. So this enzyme, this reaction is carried out or catalyzed by your ornithine transcarbamylase. Okay? So pagsasamahin lang natin sila. Okay. The next step, step number 2, is we add here your aspartate. Okay? So yung aspartate dito, saan ang galing ito? Okay. So yung second step natin is the papasok yung aspartate sa eksena and magko-condense din sila. From the citrullin, it will condense with your aspartate. So magme-merge yan. Okay? So dito sa portion na to, ito, yung nitrogen dito will react with the carbon. Okay? To produce this bond, the CN bond. Okay? And in order to carry out this reaction, you need ATP. So ATP hydrolysis will power the reaction. So the yung energy na na-release ng ATP, this will allow the reaction to proceed. Okay? So that's why we have the formation of AMP, adenosine monophosphate, and your pyrophosphate. So ano nangyari ay maproproduce natin dito yung argininosuccinate. Okay? So argininosuccinate. Okay. Step number 3 is, by the way, it's yung enzyme natin ay synthetase. Kasi in our discussion of enzymes, when you produce another covalent bond with the aid of ATP hydrolysis, you use a synthetase enzyme. Okay? So the name of the enzyme is argininosuccinate synthetase. Step number 3. So step number 3 is to we will have here cutting. So mayroon tayong makakat na bond. Okay? So this portion of the argininosuccinate will be cut. Itong part na to. So this portion will become your fumarate. So saan natin na-encounter itong fumarate na to? Fumarate is one of the intermediate of your Krebs cycle. Yan. So pwede rin siyang gamitin sa Krebs cycle. So dito, dahil nagkaroon tayo ng cutting, mapoproduce natin yung arginine. So arginine is our amino acid. Yan. So amino acid natin yan. So this reaction is catalyzed by your argininosuccinate lyase, or simply known as your argininosuccinase. Okay? So what happens now with your arginine? So for the last step of the urea cycle, meron tayong ditong action ng water. So with the aid of water, so ito ay hydrolysis. So the water here will cut this bond. Okay? So that will cut this bond, and dito na mapoproduce yung ating urea. The remaining part of the arginine will become your ornithine. So ornithine is the first step nga natin doon sa UV cycle. So na-recycle natin siya. Okay? Na-recover natin siya. Okay? So the ornithine is now ready again to accept another carbamyl phosphate, and the urea cycle will go on. Okay? So ma-excrete na natin dito yung urea. So, yung urea natin dito, paano ba siya naging ganyan yung structure? Okay. So, yung isang portion dito ay saan galing? Isang portion ay galing sa glutamate, 'di ba? Nung ang glutamate natin ay nag-undergo ng oxidative deamination, nagkaroon tayo ng NH4. Okay? Tapos saan yung sunod? Yung C double bond O dito, galing yan dun sa inyong carbon dioxide. And the last one, this NH2 portion comes from your aspartate. Yan. So, yung portion ng aspartate. Okay? So, aspartate. Balikan ko pala yung step kung saan pumasok sa eksena yung aspartate natin. Kaya nandito yung malic aspartate shuttle kasi 'di ba? Ang yung portion na yung yung first two steps kasi natin ay nasa na? Sa localization, ito. Yung aspartate kasi natin ay sa cytosol. So, cytosol yan. So, para mapalabas natin yung mapalabas natin yung makaproduce tayo ng aspartate in the cytosol, kailangan natin munang i-convert yung oxaloacetate from the matrix. So, yan. Oxaloacetate, magkakaroon tayo dito ng transamination reaction, mapoproduce mo yung aspartate. So, yung aspartate na yan ay pwede mo siyang palabas ng cytosol. Yan. So, available na yung aspartate, saka, yan. Pag available na siya, pwede na siyang mag-condense with your citrulline to produce your argininosuccinate. So, that's the relevance of the malate aspartate shuttle. So, we need to produce this aspartate in the, yan, kailangan siya nasa cytosol siya. Kasi, yung reaction nga kung saan yung aspartate ay nag-react with your citrulline via a condensation reaction, ito ay nasa cytosol siya. Okay? So, the net reaction is, you have your NH4 that comes from your glutamate, reacts with carbon dioxide, you need 3 ATP, you need water, and you need aspartate. So, the aspartate here is the donor of another NH3, the amino group. So, at the end of the cycle, you will have the formation of urea, 2 ADP, an AMP, and then 4 inorganic phosphate and your fumarate. So, your fumarate, pwede natin siyang gamitin for other metabolic pathways. So, mag-discuss naman tayo ngayon ng mga ibang mga sakit kung saan, ano ba yung mga pag affected yung enzyme natin, magkakaroon kasi ng sakit. Okay? So, yung first na relevant dito is yung tinatawag natin na medical condition. You call that your hyperammonemia. So, hyper means madami o mataas. So, mataas yung ammonia content sa inyong blood. Okay? So, we have type 1 and type 2. So, hyperammonemia refers to the high levels of ammonia in the blood. Maaring ito ay cause ng two reasons. The first one is a liver disease. Kasi nga, di ba, it happens in the liver. O kaya naman ay yung ating enzyme dito, yung carbamol phosphate at carbamol phosphate synthetase natin ay impaired. So, kapag impaired do, hindi tayo makaka-produce ng carbamol phosphate. So, anong nangyayari ay, naiipon yung NH4 sa blood. And that will have a profound effect dun sa ating blood. So, magkakaroon tayo ng tinatawag na alkalosis. Okay? So, kapag, yung bang enzyme naman natin ay affected din. So, like, for example, dito sa citrulline and condensation with aspartate. So, pag wala yung enzyme ng citrulline and aspartate, yung mag-merge sila, yung ating argininosuccinate synthetase, so, ang nangyayari ay nag-iipon naman si citrulline. So, ito naman yung tinatawag natin na citrulinuria. So, naiipon yung citrulline. Okay? The next one is what we call yung impairment ng enzyme kung saan ako-convert yung argininosuccinate to arginine. So, this one is your argininosuccinase. So, meron tayo ng argininosuccinic acidemia. Kasi itong succinate natin, yan ay, remember, ito ay may mga COO minus yan. So, yun ay mga acidic na mga compounds din. So, pag naiipon yung argininosuccinate sa blood din natin, sa katawan natin, na tumataas yung pH. Nagiging acidic yung ating blood. So, that is what we call your acidemia. And the last one is the impairment of your arginase enzyme. So, this is what we call your arginase deficiency. So, kapag may deficiency dito sa arginase, mahirapan yung tao na mag-release ng urea. So, naiipon din tong arginine sa body natin. Okay? So, ito yon yung hyperammonemia. So, now we're done discussing the fate of your amino group. So, via the 3 steps, yung ating transamination, and then the oxidative deamination, and the last part is the urea cycle. Now, how about the remaining part or the remaining structure of your amino acid, the carbon skeleton? So, the carbon skeleton will also undergo a series of reaction or series of pathways na pwede siyang magamit as source of energy o kaya naman as a precursor or starting material for your glucose. So we're done discussing this, the disposal of nitrogen atom as urea. So ito naman ang puntahan natin, yung carbon skeleton. Okay. So this carbon skeleton can be used as source ng acetyl-CoA natin. And the acetyl-CoA here will be used to produce your acetoacetate. So, kung natatandaan nyo, ito yung sa ketogenesis part. So, mapoproduce yung mga ketone bodies natin like acetone and acetoacetate and beta-hydroxy butyrate. O kaya naman, your acetyl-CoA will enter the Krebs cycle. So, upon entry with the Krebs cycle, ma-degrade siya, ma-release yung carbons natin na pumasok. Di ba kasi nga, ang acetyl-CoA is 2 carbons, this will be released as carbon dioxide. Also, yung ating portion, di ba kanina sa ating transamination reaction, meron tayong couple kung saan nakaka-produce tayo ng pyruvate. If you remember, alanine reacts with alpha-ketoglutarate. This will result to the formation of pyruvate and then the glutamate. Okay? So, yung glutamate dyan, pasok yan sa, yan yung magpunta sa oxidative deamination, tapos urea cycle. Yung pyruvate na na-produce dito sa transamination reaction, yan ang pwede natin gamitin for the synthesis of your glucose. Okay? And the last part is yung itong portion na to, like dun sa transamination reaction ng ating aspartate, di ba? If you recall, our aspartate reacts with your alpha-ketoglutarate using the transamination step din. This will produce your oxaloacetate and glutamate. So, yung ating oxaloacetate dito, yan, isang intermediate, siya ay pwedeng pumasok sa Krebs cycle. Siya ay isang intermediate. So, siya ay yung starting, di ba, sa reaction number 1, it's the condensation of your acetyl-CoA with your oxaloacetate. Okay? So, pwede yan yung oxaloacetate sa Krebs cycle. Okay? O kaya naman, your oxaloacetate, na-discuss natin ito doon sa ating gluconeogenesis, pwede natin siyang gamitin as precursor for glucose. So, kung natatandaan nyo pa, ang oxaloacetate natin, pwede natin siyang lagyan ng, pwede nating lagyan ng phosphate dun sa para maging phosphoenol pyruvate. So, yung oxaloacetate natin, kung natatandaan nyo pa yung gluconeogenesis natin, it will become PEP and then saka siya yung gluconeogenesis, then we have your glucose. Okay? So, dito, hindi ko pa ata ito na-discuss sa inyo, meron tayo ng tinatawag na glucose-alanine cycle. Okay. So, the story goes like this. Your muscle proteins will be degraded into amino acids, and those amino acids will, yung series of steps natin ng transamination and oxidative deamination. So, meron tayong nare-release na NH4, and yan na itatanggapin, yan ay mako-concentrate sa glutamate. Okay? So, yung glutamate natin, siya yung parang ini-impok, sa kanya ini-impok yung mga NH4. So, napo-form yung glutamate. We already discussed this part. This glutamate will react with your pyruvate via a transamination reaction to produce your alpha-ketoglutarate and your alanine. So, this alanine will be, yan naman ay soluble naman yan, di ba? Amino acid naman yan. So, yan ay may papadala doon sa ating liver. So, doon sa liver, yung transamination reaction kasi natin ay reversible naman yan. Okay? So, yung alanine dyan, it will react with the alpha-ketoglutarate, mapoproduce natin yung pyruvate. Okay? So, mapoproduce yung pyruvate, and then you have your glutamate. Yung glutamate, mag-undergo to ng oxidative deamination, mapoproduce yung NH4, then papasok siya sa urea cycle, and then i-release natin siya as urea. Yung pyruvate, gagamitin siya as starting material for production of your glucose via the gluconeogenesis pathway. So, yung glucose na yan, pwede mo siyang ipadala, then this glucose will be the source of energy in your muscle cells. Okay? So, that is what we call your glucose-alanine cycle. So, yung carbon atoms natin ay nade-degrade siya. So, carbon atoms are degraded amino acids. Carbon atoms of degraded amino acids are major metabolic intermediates. So, merong dalawang possible na puntahan yung ating carbon skeleton. So, the first one is the glucogenic amino acids, and the other one is the ketogenic amino acid. So, from the name itself, glucogenic, so ito ay for the synthesis ng ating glucose. So, ito. It can serve as glucose precursors via the gluconeogenesis. So, yung mga glucogenic amino acids natin na yan, ay dinedegrade yan. They are degraded to the TCA cycle intermediates. Okay? And yung second classification is what we call your ketogenic amino acids. So, from the name itself, keto, ito yung napoproduce natin. Ang napoproduce natin dito is yung mga fatty acids natin. And we can also use some amino acids as starting material or precursors of your ketone bodies like your acetone, your acetoacetate, and your beta-hydroxybutyrate. Okay? Also, this ketogenic amino acids, kapag na-degrade yung mga amino acids natin na yon, ay nakaka-produce sila ng acetyl-CoA. So, this acetyl-CoA, pwede nga siya na gamitin for production or synthesis of your fatty acids. Pwede rin naman siya pag sobra-sobra dun sa ating ketogenesis. So, makaka-produce tayo ng ketone bodies. O kaya naman ay pwedeng gamitin yung acetyl-CoA for the production of energy. So, a source of energy. Okay? So, among the 20 amino acids natin, here's the classification. So, only 2 amino acids are ketogenic. So, that's your leucine and your lysine. And we have, may amino acids kasi tayo na both glucogenic and ketogenic. So, we have both glucogenic and ketogenic, your alanine, glycine, cysteine, serine, threonine, tryptophan, phenylalanine, tyrosine, and isoleucine. While your glucogenic amino acids, ito yung magagamit for the production of your glucose. You have your proline, arginine, histidine, glutamate, glutamine, aspartate, asparagine, methionine, and your valine. So, medyo mahirap siyang i-visualize, but we can summarize that using this diagram. Okay? So, yung mga amino acids natin, pwede silang maging intermediate ng ating Krebs cycle. So, like this one, yung arginine, glutamine, histidine, proline, pwede silang ma-produce, ma-convert into glutamate, and that glutamate, di ba, tinatanggal lang natin yung NH4 via the oxidative deamination, so makaproduce tayo ng alpha-ketoglutarate. So, medyo complex na tong cycle, yung complex na yung info nito. So, this will just give you an idea na yung ating Krebs cycle ay yung tinatawag natin na ginagamit yung mga intermediates as precursor ng ibang organic compounds. Tapos pag siya naman yung nangangailangan, so pwede naman sa kanya mapunta yung ating mga pwedeng ma-produce yon using your amino acids. Okay? So, in this pre-recorded lecture video, na-discuss natin yung overall nitrogen metabolism natin. So, nag-start tayo with the nitrogen cycle. So, yung mga steps doon kung paano na gagamit yung nitrogen gas, the N2 gas, na napupunta siya doon sa bodies of your plants and animals. And then, the idea there is na produce yung amino acid from the nitrogen. Tapos, na-produce yung ating amino acid, na-synthesize yung proteins. Yung proteins na yon ay pwedeng kasama sa diet. Tapos, sa diet, yung proteins na yon ay pwede mo siyang i-metabolize. So, when you need to degrade your proteins and your amino acid, then you have several ways on that. And then, kailangan ay ma-maintain natin yung body proteins natin. So, we either degrade pag hindi na kailangan, kapag non-functional, kapag kailangan na, saka kayo mag-synthesize. And also, part sa discussion natin ay kasama yung synthesis ng ibang nitrogen-containing compounds like your precursors for nucleic acid. And then, we can also use your amino acids as source of energy. Dito sa discussion natin ng carbon skeleton as energy source. O kaya pwede natin siyang gamitin na pang biosynthesis ng iba pang mga biomolecules. And then when we want to get rid of the nitrogen atom in the form of waste products natin, so yung urea, yung ating uric acid, o kaya naman ay in the form of ammonia. Okay? So na-discuss na din natin dito yung paano yung proteasome ubiquitin mechanism, kung paano na-degrade yung mga non-functional na proteins. And then we also discussed the dietary proteins, paano na-absorb, na-digest, and tina-transport. And majority happened kanina is yung ating catabolism or the degradation. So the degradation of your amino acid, we have the two parts. Your amino group will be disposed as urea, and then the remaining structure, the chemical structure of your amino acid will be your carbon skeleton. So the carbon skeleton can be used as a source of energy or source of other intermediates natin. Sa degradation o disposal natin ng nitrogen atom, kasama doon yung transamination, oxidative deamination, and the discussion on urea cycle. Okay? So we also discuss the step by step, and yung mga medical conditions that arise from the impairment of one of the enzymes of your urea cycle. Okay? And the last part is the discussion of the breakdown of the carbon skeleton for the production of other biomolecules, a source of energy, and ano yung mga possible fates ng ating mga amino acid as intermediates of your Krebs cycle or other intermediates of other pathways like itong acetyl-CoA. Ito ay part nung sa ating lipid sa ketone body formation natin or the ketogenesis. Okay? So that ends my discussion for the nitrogen metabolism. And thank you very much for listening.